We are beginning day 3 of our EOC review. And in day 3, we're going to be looking at how cells divide. We're also going to be looking at how cells use their DNA. So this first part here, analyze how cells grow and reproduce in terms of interphase, mitosis, and cytokinesis. For us, this was actually in section 2.3. This is when we talked about cell division in section 2.3. So when you go back to your notes and you look at when does the duplication of DNA occur and what do these things represent, this exact diagram is there in your notes. Remember, cells are going to grow and get bigger. So G1 represents the initial growth. S is when we synthesize or copy the DNA, and G2 is growth 2. Then we can actually undergo mitosis, which is the division of the nucleus to get 2 identical cells. And so if you remember, we actually talked about I, little I, P, mat, C. Interphase, prophase, metaphase, anaphase, telophase, those are mitosis. And the C is for cytokinesis. And so we talked about how mitosis is just the division of the nucleus, but the cell cycle includes interphase, preparing for division, and cytokinesis, dividing the cytoplasm. So you should be able to complete the rest of this part. If you will actually, with me, skip down. Oh, I skipped too far. And look here, explain the role of meiosis in sexual reproduction and genetic variation. This is also from section 3.2. This is when we talked about mitosis makes identical cells, and meiosis preps for sexual. Remember, mitosis, the whole point is that mitosis creates identical cells and meiosis creates what are called haploid cells. So you should be able to use your notes to be able to fill this part out as well. I do want to spend some time talking about the sources of genetic variation. Remember, I'm not filling a lot of this out with you because it is directly in your notes. However, when we talk about the sources of genetic variation, we do want to know that these are the 5 sources. Crossing over is when we exchange versions of homologous pairs, and we get new combinations, new combos. Random assortment of chromosomes is during meiosis, we divide into haploid in different ways. Meaning sometimes when an egg divides, all of the ones from its mother goes in one type of cell, and all of the ones from its father goes in another type. But sometimes when we undergo random assortment, they actually get mixed up with each other. Gene mutations are going to be errors that happen during replication, and these can change the gene, and we might end up with a different protein. Non-disjunction is where the chromosomes don't separate properly, and we end up with the wrong number. And then fertilization, this should be what we call random fertilization, because because a random sperm can fertilize a random egg. A woman will make hundreds of different types of eggs, and a man will make hundreds of different types of sperm, and we don't know which one is going to fertilize the egg. I also want to go over this picture with you because this shows us a diploid cell. And so when we talk about gametes, remember gametes are going to be haploid. So if this cell has 4 chromosomes, a haploid is only going to have 2. Now the question is, does it have 2 T's and 2 S's? And that would be incorrect. Each gamete needs to have half of the information. So it could be that they have a big T with a big S, or they could have a big T with the little s. But there's actually 2 other combinations with the little t. It could be that the little t is with one type of the s, and the little t could be with the other type of the s. The important thing here is that if you look at all of these gametes, they're haploid, and they have half of the t's and half of the s's. And so that's where we started talking about cell division and how cells divide. We do want to talk in this section also about DNA. And so if you'll scroll back up and we look at 3.1.1, explain the double-stranded complementary nature of DNA as related to its function in the cell. Well, the interesting thing here is that here you have a picture of DNA. I'm sorry it's on its side. That does make it kind of hard to see. That didn't work. But if you look here, remember that the backbone of DNA is always sugar and phosphate. Below is a strand of DNA. DNA in the cells exists as a double helix. What else needed to be added to the strand to make it a double helix? Give the nucleotide sequence. So this is one side of DNA. That means that across from the A would be a T, and across from the G would be a C. Remember, these little dots represent what are called hydrogen bonds. Hydrogen bonds are weak, and they hold together DNA. So you should be able to finish listing out the base pairs. Remember, these are complementary base pairs. And so if this is DNA, you should be able to complete it and tell me what type of sugar DNA has. For example, DNA has what's called deoxyribose sugar. RNA just has ribose. DNA has T, A, C, and G, but RNA does not. RNA has different nitrogen bases. And so you should be able to fill this table out, and you should be able to tell me why it's important that these bonds are weak. There's actually 2 reasons. One reason is so that we can unzip for replication or copying. And the other reason is to unzip for and I'm not going to finish that. Do we use DNA during transcription or do we use DNA during translation? You should be able to tell me that. So we're going to go ahead. We are leaving some blanks, but that's because you should be able to go back and figure them out. We are going to go ahead and talk a little bit about protein synthesis. For example, if the strand of DNA above undergoes transcription, well, that might be a little hard. Let's write what that strand is out so that we're all sure that we know what the strand is. The strand is going to be GACCGA. There we go. If this undergoes transcription, what will the sequence of the mRNA be? Remember, mRNA is going to use uracil. So our base pairs are now A with U and G with C. So G will base pair to C, A will base pair to U, because I can't use thymine in RNA. C will base pair to G, C will base pair to G, C, U. And so that is what happens if the DNA undergoes transcription. After translation, what would the sequence be? So this is where I need to find CUG, and that will give me one amino acid, and then GCU, which will give me the second. Go ahead and cross out this part that says read from right to left. So this is where we're reviewing how we use the DNA code to make a protein. And so you should be able to complete this part. Describe how the proteins code for traits. Remember, proteins are going to be made of amino acids in the correct order. We have to get those amino acids in the correct order to make the correct shape to do the job. So for example, if you are an enzyme, we need that enzyme to be the right shape so that the enzyme can do its job. If the enzyme is the wrong shape, then it won't give us the same trait. And this is how we have different versions of trait, because some people's DNA is different, they get different proteins, and the proteins do different jobs. Explain how mutations can result in interactions in the environment, can result from interactions with the environment. So we do want to know that mutations can sometimes be caused by the environment. Like radiation and chemicals. And so you should be able to name the two types of mutations, which type is likely to have a greater impact. That's directly in your notes. This is going to be found in section 3.2 in our notes. We already talked about this section here. And so now we're going to go on and talk just a little bit about genetics. We're going to cover a lot more of genetics in class, but what we want to pay attention to is all of these different inheritance patterns. Mendelian dominance, incomplete dominance, codominance, sex linked, and multiple alleles. And so you should be able to use the word allele. For example, here, this is the dominant allele, and this is the recessive. Dominant, recessive. Give the genotype for the parents. This parent, they're both heterozygous, which means they have the dominant version and the recessive version. Because they have the dominant version, they only show the dominant physical appearance, which means that these parents, let's say pea plant parents, would be tall. The genotypes and phenotypes of the offspring, remember genotypes are going to be written as 2 letters. So the genotypes of the offspring include big T, which is tall, big T, which is tall, and little t, which is short. And so remember that we assign these letters to represent versions of the DNA gene. And then we look at what the parent has, and we see how could those parents possibly combine. This is a really important question. What environmental factors might affect the expression of these genes for height? Some genes produce intermediate phenotypes. Here it's asking about cross a pure breeding red with a pure breeding white. We do want to notice that here we're actually talking about incomplete dominance. And so you see that little R prime symbol that we use for incomplete. The next part of the notes actually talks a little bit about different diseases that we have. So you should be able to talk about the different diseases that are found. No, sickle cell, cystic fibrosis.
Alexis, stand back, meet your right out front immediately. Alexis, stand back. Teachers, if she's with you, please call the front office.
To work the blood type problem, you are going to have to write your parents' alleles off to the side. If the woman has type A, she has to be IA as her first letter, and the man has to be IB as his first letter. But notice their first child is type O, which is little I,
Students,
little
may I
I.
have your attention, please? Those of you who are interested in the FCA celebration service this afternoon, it's beginning in 2 minutes in the auditorium. Please make your way there now.
The final version that we want to practice are sex-linked traits, and these traits are on the sex chromosomes. The male sex chromosome is Y, and females have the X. But remember, females are going to be XX, and males are going to be XY. Remember, sex-linked traits are only on the X, which means that a guy might have a letter on the X, but never an allele on the Y. So we have a female who is a carrier for hemophilia. Remember, hemophilia is going to be recessive. So a female who is a carrier, she does not have hemophilia, so she gets the dominant allele, but she is a carrier. She's hiding it. She gets a recessive. And then the male is normal. He gets a big H on his X, but he also gets a Y with nothing on it. So you should be able to fill out the rest of the Punnett square. So lots of information in that section of notes. Real quickly, just to finish it up, we do have this last, there's actually almost 2 pages, but you should be able to do the karyotypes. Karyotypes are in section 3.4, so you want to go back and read about karyotypes. Remember, these are pictures of the chromosomes. And then you want to also look at pedigrees from section 3.4. Remember, we do have our important questions. Number 1, does it skip a generation? If it does, it is recessive. So remember, in pedigrees, here we have 2 parents that don't have it, but their child does. This means that it is recessive. However, when we look at this, we also see a daughter who has it. Our second question is, does a daughter have it? If a daughter has it, in order for it to be sex-linked, her dad would have to have it as well. Since her dad doesn't have it, it has to be autosomal. It can't be sex-linked, because in order for a daughter to have it, she had to get the X from her dad, and that would mean that he has to have it as well, and he doesn't. In this section, it was a long one. There's also some information here about different expression of traits, the DNA fingerprinting, and transgenic organisms. And so we'll go over some of this information in class, but I would like for you to do your best to work all the way through, finish all of this as well. It should be pretty easy because it should be directly in your notes.